Right. So, yeah. So for the rest of these two seminars, we'll be talking about the computational analysis of this type of data that comes out, the data that you get off your sequencing machine. And the first part is really about pre-processing and quality control. And if you ever worked with genomic data before, you'll know that it can be very noisy. So this part is actually very important. And if you don't get this right, there's a very good chance that you will be analyzing technical noise and other types of artifacts and mistaking those for for real biological signal. So, fortunately, at this stage, there is the general workflow for how to process this data is very well established. So here is a very nice overview from a recent review. So as I told you, that sort of the first part is really about pre-processing and doing various types of filterings and normalizations. And then the second part is really what you refer to sort of more biological type of analysis, where you do the type of study where you can sort of try to draw biological conclusions and inferences. So yeah, so the first thing you need to do is really just to look at your reads and do quality control of those. So this is no different from any other sequencing experiments. So you can use whatever type of favorite tool you have for that. I'm not going to go into too much detail, but I'll just show you sort of one small example here from a study which I was involved in. And so what you can see here is that this is an output from FastQC, which is a popular tool. So you can see here is that you have sort of somewhat lower quality at the beginning of the read and at the end of the read, which is sort of fairly common. What's surprising here is that we have this unexplained dip right at the middle of the reads. We have no idea where it came from. In this case, we sort of decided that it's unlikely that this is going to have a major impact on the mapping or anything else. So we just decided to keep all the reads. But this is sort of important to look at in order to make sure that you're happy. Then, of course, there can be various types of adapters and so on that they have put on for the experiment. So you need to be able to trim those off as well. And there are also softwares for doing that. So the next step that you need to do is demultiplexing. So that's basically identifying which read came from which cell. So if you're using one of these sort of low throughput experiment where you typically put, like for example, SmartSeq2, where you put the cells in different wells here, then you would also know sort of which barcode for each cell that was put in each well. For the droplet-based methods, this can be more complicated. So you might have to do a little bit more work for the demultiplexing. And it should be said that in sort of if there had been no errors, this would of course have been straightforward in principle. But what we also have to take into consideration is that we can have sequencing errors on these barcodes. So we need to account for those when we do the demultiplexing. So here is a sort of an overview of the type of a schematic overview what you get out from your experiment once you've done the demultiplexing. So this is for a droplet-based type of experiment where you also have this unique molecular identifier, the UMI. So here we've been able to group each read by the cell, which was this barcode that was unique to each cell, and then we have this UMI. So now I'm going to tell you next about what the UMI are. So UMI stands for unique molecular identifier. And it's basically a short tag that you attach to each of the transcripts prior to amplification. So this allows you to sort of distinguish the reads that came from, so if they came from the same molecule, the same initial transcript, then they will have identical UMIs. But if they came from different transcripts, then they will be different. So this is a way of sort of very efficiently removing PCR duplicates, but it also allows you to sort of do absolute quantification because you know that each UMI will correspond to one transcript. So here is sort of illustrated how useful these can be, and this is a very small schematic example. So here, for example, the red read got sort of amplified 4 times. And so if we then went ahead and did sort of variant calling here, you might assume that you would have 4 of the A's versus 2 T's, whereas in reality it should actually be only 1 A and 2 T's here. And that conclusion you'll be able to get if you have the UMIs and if you collapse the reads that corresponded to the same UMI. So UMIs can lead to trouble again because you can have various types of errors. So one error that can happen is if you have very abundant transcripts, and you'll often have quite short UMIs that can be as short as 6 base pairs, for example. Then there is a risk that you'll have collisions. So you'll have initially it was actually different transcripts, but because the pool of UMIs was relatively small, they will end up getting the same UMI, and then you will think that they were PCR duplicates when they weren't. Another issue is of course that you can have sequencing errors in your UMIs. So then you might think that things that were really different, things that actually came from the same transcript actually instead are different because of these errors. And you can, but fortunately, there are many different tools available for dealing with these types of problems. One of them is called UMI tools, and it is based on using a sort of a graph approach. So basically what they do here is that they look at sort of nearest neighbors in terms of the number of errors or differences in the Hamming distance of these UMIs. And then based on the patterns, they can sort of infer what types of errors were the most likely to have occurred. So here is a fairly common example, a fairly common situation, which is when you have one node which has a very high number of counts. So that's presumed then to be the sort of the originating UMI, and then you'll have that surrounded by nodes with much smaller counts, and that is then likely to have been caused by having at some point during the amplification process you had an error, and hence you have fewer reads mapping to those UMIs. So if you take this into consideration, then you can sort of prune this and basically merge the UMIs that were sort of caused by this error process, and so that will allow you to avoid those types of errors. So once we've sort of collapsed our UMIs, and so then we need to sort of align the reads and quantify them. And here there are really sort of two main strategies. The first one is to sort of use the traditional route where you would use a mapper such as star to align the reads to the transcriptome, and then you can use for example, feature counts or HTC to just quantify the number of reads that you have for each gene. The other method, which is very popular when you have these very large data sets, is to use a pseudo aligner instead, which, for example, Salmon and Callisto are the two popular ones. And I would say that that's probably the most popular strategy these days. There are also methods that can sort of are specifically designed for these droplet-based experiments to do the UMI analysis and the quantification. So Alevin is one such method from Rob Patro's lab. So so they really sort of try to model the process of these droplet-based methods and sort of try to do clever things in order to reduce the errors from this UMI deduplication and the quantification. And this should hopefully lead them, allow them to sort of rescue some reads that would obviously have otherwise would have been hard to map, for example, due to multi-mapping. So this is sort of illustrated here in this figure from their paper, where they compare against Cell Ranger. And as you can see here from their genes where reads are uniquely mappable, you don't really have much of a difference. The difference really happens here towards the left end, where you have these reads, genes that are sort of probably arose due to duplication events. So there are a lot of reads that are areas that are multi-mapping. And here their method is able to do much better. So presumably you get better quantification of these genes, whereas here it doesn't matter very much. So So now that we've sort of understood what our UMIs are doing, and we've counted those and sort of mapped to the reads, to the genome, then we can sort of start quantifying the different genes. So now I've extended this schematic that we had here before, so we can also sort of see how many counts we have for each different gene in each of these different cells. And so the next step to do is it requires us to take a step back, and this is again really specific for the droplet-based methods, and to sort of consider an important aspect of the sort of experimental design here, or how you carry out the experiment. So remember, the goal is to put one cell into each droplet, and this is, of course, the cells are very small, and so are the droplets. So this is not as trivial as you might think. And what happens in practice is that you end up with a Poisson distribution of the number of cells in each droplet. So you want to sort of, so now you need to strike a balance that on the one hand, you can think, that's basically something you control when you put your cells into the machine. The number of cells controls the rate of this Poisson parameter. And if you put too few cells into the machine, then you'll have a very low rate. So the problem then is that you'll end up with a lot of empty droplets, and that is basically a waste of these reagents. So that means that you'll get sort of fewer cells out of your experiment, and it will cost more. The other problem is the other direction is not good either, right? Because then you put in more cells, and then you end up getting more doublets, and those can really sort of screw up your analysis, because it's not obvious a priori if you had one or more cells inside a droplet. So we need to strike a balance here, and of course we need computational tools in order to be able to identify these problems of empty droplets or droplets containing multiple cells. So this is what I'm going to be talking about next. So the way you can think about this empty droplet detection sort of conceptually is very straightforward. So basically you just count the number of unique UMIs that you found in each droplet, and then you'll rank them. And what you're really looking for is some sort of a big drop where you, and the hypothesis then is that big drop would correspond to a situation where you go from having cells, viable cells with UMIs, to having these empty cells where you just have sort of various type of debris that somehow got encapsulated. So this is a very typical situation, that you'll have a sort of a few cells with a lot of UMIs in them. And that those might correspond to doublets, and then you'll have a sort of a long range here, and then you'll have some sort of drop here at the end. And this is sort of this drop where you need to sort of decide where to do your cutoff. So the most straightforward way is just to sort of do a simple thresholding. There are, of course, more sophisticated methods for doing that. One example is this empty drops method, which sort of uses it like a Dirichlet model to try to build a model of the background distribution, and that allows them to sort of look at the contents of each cell or each droplet and sort of compare it to this presumed background. And if they look very different, then they can sort of rescue the cell and not throw it away. So that allows them to keep some of these cells that have lower counts. Whereas the threshold method would just sort of drawn a line here and thrown away everything to the left. So that is hopefully a little bit better. The other problem that I was telling you about is sort of doublet detection, and this is where these dual species experiments that I talked about earlier are very useful. When you do those types of experiments, you're hoping for this type of plot, which is shown here, which is known as a barnyard plot. So, but what you're basically hoping is that you would get one cell in each droplet, and then you can map those reads that you get from each droplet to both the human and the mouse genome. And then you would, of course, hope that you get a lot of matches to one of the genomes and no matches to the other. And this is sort of what you see here when you have the points sort of hugging the X and the Y axis. But then, of course, you have a few points here on the diagonal, and those presumably corresponds to these multiplets, where you have sort of one cell from human and one cell from mouse. So this type of experiment you'll often see when people develop new technologies, and it's considered an important benchmark. Now, of course, in most practical experiments, you won't have two species to deal with, but you'll only have cells from one species. So then you'll need a different type of approach. And again, there are multiple methods to do this. Two of them are called Doublet Finder and scRNA, and those and others, they use pretty much the same strategy, which is basically that you take your cells, and again, you assume that the number of doublets is very small. And so what you do then is that you create artificial doublets. So you take two cells and you create the doublet, and then you analyze this sort of new sample where you have introduced these artificial doublets, right? Which, of course, you know which ones they are. And what Doublet Finder does is that it then builds the nearest neighbor graph, and then it takes the cells from the sort of original sample that are look most similar to these artificial doublets. It will assume that those are actual doublets as well, and then it will sort of throw those away. So this might sound like a bit of a circular reasoning, but it seems to work quite well in practice. So another type of problem which is sort of related to this is called ambient RNA. So ambient RNA is basically you'll also hear the term the soup. So that's RNA that somehow gets into all of the droplets. So it has presumably leaked from some cells that were in your samples, and then they've been able to enter all of the droplets. And now they will sort of contaminate the samples by sort of raising the background levels across the board. So this is obviously something that you want to get rid of so that you don't think that you have these transcripts everywhere, because it can mess up with your differential expression analysis. So the first method that tried to correct for this background and identify the ambient RNA is called Supex. And what they do is that they look at the very far end of this tail here of the droplets that contain the fewest UMIs, and then they assume that corresponds to the soup, and then they sort of build a model of the soup, and then they try to sort of subtract it from the other cells. I think the main drawback of this method is that it requires you to input a list of genes that should not be present in each of the cell types that you've found. So that sort of can really raise the bar in making this more complicated to apply this method. So there are other methods out there. I think the most recent one, which a lot of people seem to be using these days, is called CellBender. So this is based on neural networks. It uses a deep autoencoder. It basically allows it to it will build a model of both of the noise and of the signal, so it can detect both ambient RNA and doublets. And as you can see here, it seems to be doing a really nice job of sort of basically removing these types of sort of low-level background reads and to push the background almost all the way down to 0. So now basically what we've done is that we've from our droplets, we've removed the ones that were empty, we've removed the ones that were doublets or corresponded to multiple cells, and we've also removed this sort of background soup. So now we hopefully only have droplets left with individual cells in them, but we still need to do another round of filtering. And it's basically to try to sort of remove low quality cells. So this, what people typically do here is that they look at sort of 3 quantities. So one being just the number of reads, the other one being the number of genes detected, and the third one being the fraction of mitochondrial fraction of reads mapping to the mitochondrial genome. So the reason for looking at the last bit is because it's basically if cells are sort of dying, then they will typically release a lot of mitochondrial DNA. So this is really an indicator of a cell which is sort of stressed or is problematic in various ways. Unfortunately, it's sort of hard to, there are no sort of strict rules for what is sort of considered an outlier in terms of the number of genes or the number of reads. It's really something which depends on your sort of on your platform, on your sample, and so on. And so there are no hard rules. Really, what you have to do is it's more of an art than a science, and you have to sort of, the goal is to sort of remove outliers, and you're looking for things that are sort of reasonably Gaussian. And so that's what you really need to strive for here, to get the nice uniform shapes. And again, you want to remove on both ends, not just at the bottom end, but also if you have outliers with very high counts, that they can also mess up your analysis later on. So yes, I think I will pause it here